So the topic of this video is active transport, kind of the basic concepts. But before we get into active transport, let's review a few things. On the left, let's review diffusion. Notice there's a high concentration of molecules on top, a low concentration on the bottom, and because of diffusion, molecules move from a higher concentrated area through the cell membrane to a lower concentrated area until there's an equilibrium. In the middle, we have facilitated diffusion. Once again, a higher concentration on top, a lower concentration on bottom, and molecules move from the higher concentrated area to the lower concentrated area, but through the protein channels. Now, these are both examples of passive transport. They do not require ATP energy. On the right-hand side, let's introduce active transport. Notice a lower concentration of molecules on top and a greater concentration on the bottom. Well, with the help of ATP, molecules can be moved from the lower concentrated area to the higher concentrated area. Let's explore this in more detail. So with active transport, it's the movement of molecules across the cell membrane, but this time with the help of ATP, adenosine triphosphate. This is the energy molecule created by the mitochondria. This is the molecule that gives the mitochondria the nickname of the powerhouse. Now, there's a few types of active transport we're going to talk in this slideshow. The first is with the use of carrier proteins, and that's what you see in the picture, that big green thing in the middle of my picture. That's a carrier protein embedded in the cell membrane. And so carrier proteins are like pumps. You know, you might have a bicycle tire pump like this or one like this, but whichever pump you have, pumps require energy. You can't pump up a tire without using some energy. You can't pump up a flat soccer ball without using some energy. Pumps require the input of energy. And in this case, we're going to see that molecules get pumped from a lower concentrated area to a higher concentrated area. In other words, molecules move up or against the concentration gradient. In my picture, there is a higher concentration of the brown molecules on top, a lower concentration on the bottom. And so molecules like this brown molecule will load into the carrier protein, and with the help of an ATP molecule, the carrier protein changes its shape to allow the molecule to pass. And so energy from ATP changes the shape of the carrier protein, allowing the item, the molecule, to pass. Let's see that again. A molecule loads into the carrier protein. With the help of ATP, this carrier protein changes shape, allowing the item to pass. And let's see this one more time. The last brown molecule enters into the carrier protein. With the help of ATP, it changes the shape of the carrier protein, allowing the molecules to pass from a lower concentrated area to a higher. Now, here's an analogy. Maybe you're in this boat and, oh no, you just crashed into an iceberg and there's a big hole in the boat, and now water from the ocean is rushing in. You're sinking. Well, it's a good thing modern boats today have what's called a bilge. It's a water pump, and hopefully you still have electricity if your boat is ever sinking. Because what the bilge will do is it will pump the ocean water back into the ocean. This way, your boat can stay afloat long enough, either for the boat itself to get to shore or for help to come and rescue you. Now, why do I bring up the analogy of a bilge? Because nature invented the bilge, billions of years ago. This is an organism, a microorganism called paramecia. It lives in rivers and lakes and streams and in watery environments. And right there is an organelle very similar to a bilge. Basically, it's a water pump, and let's see how this works. So in this little animation, notice the concentration of the water. Let's pretend it's 96% water, 4% solutes. And so through osmosis, from a high to a low concentration, water is going to enter this paramecia, and water is going to flow into the paramecia. It's going to swell, it's going to expand, and if you're the paramecia, this could be deadly. Because if too much water diffuses into you, the paramecia, it could cause you to burst and die. And obviously you don't want that. And so what paramecia have is nature's equivalence of a bilge. A contractile vacuole is activated. It repeatedly contracts to squirt out the extra water that is diffusing in, much like a bilge does. But in this case, notice how the water is being pumped out of the paramecia cell from a lower concentrated area of 90% to a higher concentrated area of 96%. Now this bilge requires energy in the form of ATP, but it's a good example of how the cell can pump molecules up the concentration gradient. Now, here's another little analogy right here. Pretend you're this kayaker. And if you're this kayaker and you're facing the opposite direction that the river is flowing, do you need to spend energy in order to move? And the answer, of course, is yes. You have to paddle very rigorously in order to move up the river, kind of like molecules moving up a concentration gradient. Well, there's other types of active transport as well. One would be, the next one to talk about is one called endocytosis. Now, endo is a prefix that means inside. Cyto means the cell, and anything with osis in it, osis just means a process. So this word literally defines itself. It's the process where cells take something inside of themselves. Let's see how this might work. And so the process in which the plasma membrane, for example, takes in a substance. And items are often carried by a vesicle. Here comes a vesicle carrying the letter P over to the cell membrane of the cell. Now the P, pretend, stands for protein. Now this protein's too big to go through the phospholipid bilayer. This protein's also too big to go through protein channels. Well, there's another way. Endocytosis. There's another way for molecules to get inside of cells. And so at first, the vesicle fuses with the cell membrane. And then notice what's happening is that the vesicle is being absorbed into the cell membrane, and eventually the protein has been taken into the cell. And now this cell has acquired and taken in this protein. That would be an example of endocytosis. Well, here's another example of endocytosis. Well, here's a virus. Pretend this virus is infecting a person. Well, we have, of course, cells of our immune system. Here's a white blood cell of our immune system. And what the white blood cell begins to do is engulf and surround the virus. So the cytoplasm and the cell membrane of the cell literally begin to wrap around and to engulf this particular virus. Now, notice the virus has been taken into the cell. Hence why this is an example of endocytosis. But now this virus is actually not dead. It's just trapped inside of a vesicle. Well, don't forget, cells also have lysosomes. Now, I like to use animated scissors to represent what's inside of lysosomes. Lysosomes, again, have digestive enzymes. And so the vesicle will fuse with the lysosome, and when that happens, the digestive enzymes, in this case the scissors, will chop up and break down and destroy the virus, ridding your body of infection. So this is one way that our immune system works to keep us free from infection. Now, another type of active transport to mention is called exocytosis, and this is really just the opposite. Exo means outside, as we said a moment ago. Cyto means the cell, and osis is a process. So this is the process when a substance is expelled or released to the outside of the cell. Now, here we have a Golgi body. That purple object is a Golgi body with the letter P on it. Again, P stands for protein. And this Golgi body will wrap the protein inside of a protective little vesicle. And then the item, in this case it's a protein, not every example is a protein, but in this example it is, but the item is carried by the vesicle over to the cell membrane. And because the cell membrane and the vesicle are both phospholipid bilayers, the vesicle fuses with the cell membrane. And notice what's happening is that the vesicle is being absorbed into the cell membrane, and eventually the protein is released to the outside. Again, another example here. This is a good example here of exocytosis. And so there you go. There's a little quick review video on how cells can use ATP to move molecules either in or out of themselves. I hope you found this video helpful. Thank you for watching.